Welcome to a very special update from Foldit. In this video, we're taking you inside the lab here at the University of Washington Institute for Protein Design. We're recording this video in the midst of the global coronavirus pandemic. Most of our team is working from home at the moment, but some scientists are still coming in because they are working on a possible coronavirus cure. All the experiments you're about to see are the same ones that we will be conducting soon on some of the very best Foldit player designs. If you aren't familiar with Foldit's coronavirus puzzles, we've got a video on that in the description below. Right now, the team has identified 99 of the most promising Foldit player solutions. We'll begin testing those molecules shortly, but we didn't want to wait to show you what that's going to look like. The goal of these experiments is to manufacture proteins that were designed on a computer, then to test them to see if they bind to proteins on the coronavirus. If they do, these molecules might be considered candidate antiviral drugs for blocking COVID-19. We want to emphasize that what you'll be seeing in this video is just the first step of our drug development process. Much more work, including clinical trials in humans, will need to be done before any new protein drug is ready. Those crucial experiments will take time. The scientist conducting these early experiments today is Longxing Cao, a postdoctoral scholar here at the University of Washington. He's working to stop COVID-19, but before you get worried about his health and lack of face shield or respirator, there is no live virus in our lab. Instead, we use this. This tube contains a purified sample of the novel coronavirus's spike protein, which is the same gray protein you've seen in all Foldit coronavirus puzzles so far. Because we work with a recombinant protein, there is no risk of infection. Our scientists have been designing proteins against this target, just like you can do in Foldit. Because they work here full-time, they've had a head start. Our experiments always begin with synthetic DNA. DNA is the blueprint for making proteins. Thanks to new DNA printing breakthroughs, it's now possible to create from scratch thousands of synthetic genes that encode whatever proteins you want. This tube alone contains over 12,000 genes, and it costs about as much as a used car. Once we've acquired the DNA that encodes the new proteins, we need to put it into cells so that they can carry out those genetic instructions. In these experiments, we grow yeast cells. Laboratory yeast are great at making proteins. In this case, we've genetically engineered each yeast cell to not only make one specific designer protein, but to also decorate the outside of the cell with many copies of that protein. This is called yeast display. For a designed protein to block coronavirus, it has to bind to it. We measure whether this binding is happening using an instrument called a flow cytometer. This machine flows a thin stream of liquid containing the cells in front of a series of lasers. The stream is so small that each cell passes the lasers one at a time. The lasers illuminate fluorescent molecules that have been attached to the proteins. The spike protein fluoresces at a different wavelength than the binder. If the binder actually sticks to the spike, the machine will report seeing both wavelengths at the same time. That's how we detect binding. Today, we found some hits. These molecules will still need to be studied more here at the University of Washington. They may even need to be further optimized on the computer, including inside Foldit. So stay tuned for more puzzles and more updates from the lab. As always, thanks for watching, and thanks for playing Foldit.